Welcome. So today we're going to talk about comparative modeling, and it's going to be a little bit of a push. It has 145 slides, but this is not all. I'm going to go through another 160 after that. And we will do all of this before 2 hits the bell. So that means I'm going to just try to zoom. I'm going to fast forward to certain parts, and that will be partially driven by you. The reason why I do it in this way, usually those were 4 lectures, and the reason why I'm zooming so fast this time is because of the way things happen this semester. I just want to come to other parts of the lecture that I consider more important. Next Tuesday, remember, there is no lecture. This has not changed, but next Thursday, Predrag Radivoje from Indiana University will talk about disorder prediction here. And that is another reason. So Predrag got in, and the time was already limited. Okay, many slides come from Andrea, and let's begin with this set of the background. So in principle, the goal of structure prediction is in comes protein sequence, out of protein structure. Structure is determined by the sequence alone, or the sequence in its environment, in solvent. And that has been shown by Anfinsen, who got a Nobel Prize in Chemistry in '72 for that. Now, this is the oncogen KRAS. This is KRAS with the variant. Do you see a difference? So this is the native version, the wild type, and the one where the glycine 12 is varied to cysteine. Do you see any difference? The colors?
One helix just, there's one helix that doesn't actually exist in another one. No, the other one. The central helix.
The central helix here? This one? This is, okay. It's difficult sometimes to draw these things, but they are in fact 2 helices. Yeah.
The left one also a little bit faint.
This one?
The left one, the left helix.
The left helix. Yes, the left helix does differ a little bit. But, do you see any major difference? No. But the major difference is actually here in the binding site. This is the disease-causing variant in this protein. And you can, again, this lecture is entirely about protein structure prediction, but how we need protein structures to understand function. Here we have an example where essentially most of what we are going to predict over the course of this lecture is essentially the same. The difference is in the little region, and that little difference matters. There is, if you wanted to call it that way, a little bit of a giveaway here, the binding loop. In fact, that induces the change. But what we will today talk about is the difficulty of modeling. One aspect of comparative modeling is this kind of thing is exactly difficult to get. To capture that is complicated, is one essence. Now, here is the example of 2 proteins, both in human, that have 85% sequence identity. One is RAS, one is RASH. And you see, now you're beginning to see really the helices differ, but still, essentially this is a similar structure, and even the binding site is somehow still similar, although ultimately they are different function. We're getting down to the level of fly in orange. You still maintain structure. Now the binding site, it still has some similarity, but there's some other binding interface. When we get down to the purple here in the bacterium, 19% sequence identity, then we still maintain structure, but now we actually completely lose many functional aspects. There's still somehow similar binding here. But what we are trying to do is essentially capture those things. Another aspect here is, in order to model structures, one thing to look at, this is conserve. Conserve is a method from the group of Nirbental. Mapping simply the conservation and the sequence alignment upon a surface, and that captures aspects of function of binding. What I show here is 2 electrostatic surfaces of the same protein in some sense, or the same 3D structure. So what happened here, and that is Diana Murray who did this work in the lab of Barry Honig, the underlying story here is there was a structure for an archaea protein. She built a model for the human protein that relates to it. And the model essentially used exactly that structure, but the electrostatic surface looks very different. So these two images are done on 3D objects that are identical, but the electrostatic features of the underlying sequences differ so much that you immediately appreciate that they will have different function. Okay? So sometimes you can use the model of the same 3D object to learn something about function that actually is different from the object you actually modeled it from. You can use these models to sort of aggregate into larger constructs, but ultimately the idea is to somehow find which of these many different protein structures a particular sequence belongs to. The idea to do this is you start with a query here. You find in the PDB database a template, you align them by sequence, you find that template by essentially sequence aligning them, and then you essentially look up the structure from the PDB protein. The reach of comparative modeling, so how much of that can you do today? And I'm going to first show you the reach for all the known protein structures. So the entire pie that I will show you represents all known structures. So we begin, and this is an extrapolated number. That is why these numbers are not quite right. And the first one here, experimental structures, is immediately wrong. So currently, as of yesterday, we had 63 million proteins in the database, compared to about 120,000 structures, 3D structures. And you see this ratio is not 13%. So that is how much this is an estimate. Now, the reason, in fact, why, or not the reason, sorry for the animations. What did I do now? And this is what I spent my whole day on. So one other aspect here of protein structures, I say there are 120,000. This is from an older version when there were 70,000, half of that. But you immediately see that going down to 100%, so only counting every single protein, throwing out every duplicate entry, reduces it by almost a factor of 2. Why is that? Going down to 95% here, you get to more than a factor of 2. But why is the first reduction?
Different species.
100%? Do you have two proteins of different species? Do they ever have 100% sequence identity? Anybody? I have never seen that. It could happen. They can be very, very similar. And it could happen. Maybe there are cases. But it's not very likely. But this is clearly not explaining a reduction from 70,000 to 42.
Maybe it's just that some interesting proteins are measured again and again, and it's just
that is it's a very important problem, a very important point. What could be the reason? So measure it again because it's an important protein. Yes, what else could you think about? That is more, in some sense, really relevant for 3D information. Could you imagine something that you want to look at for the same two proteins in two different states? How? Is that a yes or? I don't know.
Maybe like, you know, like it's kind of measured, like doesn't it like kind of modify protein the sequence again?
No, 100%, no change.
No change, right?
Anybody?
Maybe I try to crystallize it in different conditions.
Yes, but why would you want to do that? It is somehow, it's a little bit what Michael says, right? It's an interesting protein, so maybe one condition you get it first and then you do a refined structure. And that, in fact, is one reason, because you get better resolutions. But there's something very, very important that is missing here.
Different binding partners?
Yes, substrate. So binding or not binding.
Yeah.
Okay, so in fact that is exactly one of the main things why you have structures, to understand how the structure is changing upon binding. How you from the structure can understand how the binding happens. And that is a substantial part here in this. It's not all of it, and again what both of you said, so getting different crystal conditions to get a better resolution. There's many of them. Doing it again because somebody else did it already in the direction of getting a better condition, maybe you didn't get it and you still publish it again because it's an important structure, is another one. And again, by simply seeing the same thing twice, even without binding, for important structure, that already is publishable because people care to see that, in fact, it's repeatable. So that somebody else gets a similar result. Okay? But then, you know, when we get down to the level of really different things, we are, in fact, already at a level of factor 5 reduction, right? But again, comparative modeling in principle tries to take a sequence of unknown structure, find some protein in the PDB that has a similar structure, and the simplest way of the H value, where we have the proteins that have similar structure in blue, proteins that have different structures, the simplest one, you would put the threshold in here, and then simply cut off by sequence-sequence comparison. Sequence slightly profile comparison would do something like this. Then we push a little bit further into the twilight zone here, and then we get down to some number like that. And ultimately, there's the midnight zone. Most proteins that have similar structures have no sequence identity. I will not find them. But somehow what I do find here is roughly this fraction, a little bit over half. So that means I have only 120,000 only, but I have 120,000 structures, and I can build more than 30 million models. Okay? And I can do that without a lot of effort. That's the biggest leverage of the experimental data. That's the biggest improvement that computational biology can give over experimental values, and that's the biggest impact. And that's the best prediction of protein structure. For most of the remainder, we cannot do anything, and we are essentially back to having to predict 1D structure alone. When we look at entire genomes, we see that there is a slight difference for different species, is what this says. The other aspect of comparative modeling that I like very much, and this again is from the past, when in 1991 you essentially predicted with some way of doing it this fraction, in 5 this, and 99 this, just by the growth of the database, not by improving your method. So in some sense, for me, this is a very positive outcome. If you develop a method and 20 years later it works much better, and although you didn't spend any time on it, this is a great reality of comparative modeling. So I'm not going to go into Jinfeng, and I'm not going to go into these data. Let's just begin with some of the concepts. Comparative modeling, and that's what I refer to. Historically, you will find homology modeling simply assuming that the reason why you can do that is because there's an evolutionary collection, and there's some truth to that statement. There's the target, it's the protein to model. Template, that's the protein to model from. So this is the query, and that's the experimental structure. Essentially, what you want to see is that from this target you want to predict the template, or that template, by simply copying in some sense the coordinates. The way you identify the template is simply by alignment, and the better the alignments, the better the start. Then you build the model. What's the simplest way in which you can build the model? So again, I'm sorry that I rushed through this. You pick a protein in the PDB that you say is most similar from sequence to your query. For that protein in the PDB, you know the structure. It's experimentally known. Okay? You don't know the structure for your query. You have a pairwise sequence alignment. How do you build a model? It's the simplest thing you can do.
Copy the old structure and just run some relaxation simulations.
Exactly. Well, the first part of your sentence was the simplest one. You simply copy, cut and paste, right? You take the coordinates. So the sequence alignment implies a corresponding residue, right? For every single residue you know in the known structure, what is the corresponding one? You go through it and you copy the coordinates. Okay? That's the simplest. Then what you will do is check out how well that simple model works. How could you assess the model? What could you look at?
For example, there's actually even possible. You know, like for example, if you have
Why would it not be possible?
very wide and then let's say you have a gap and then your
So one thing is that you would have a gap between regions. That could be because there are certain residues missing. Okay, we will get back to that. We will call that the loop modeling problem. So that your protein that you're trying to predict, say the human protein from a fly structure, has an insertion that the fly protein doesn't have. So that's one problem. But this is not really assessing the model. That is assessing how well the model fits to your sequence. But what, no. This is essentially, you would have, in your model, you would have missing parts. Okay, you could call that the quality of the model. Okay, I take that. But it's not quite what I meant. Anything else?
So any homology modeling approach will be based on alignment quality. So you could check known structures and see how good or bad it is.
True. But this is sort of the step before, right? In some sense, that would dictate how you do your alignment. What I'm asking here, once I have a model, and I do the simplest thing now, I do cut and paste. What kind of things could I find that would immediately indicate a bad model? Again, a not fitting model is, or not existing model is one clear criterion for a bad model. What other criteria could you think about? And I'm sorry for the way I do my presentation today. I have so far away. Okay, the next time I have to do it differently. I believe I'm just going to stay on one slide and then going to do the rest of the talk on one instead of 160. So essentially what I mean is, this purple is the experimental structure. The green one is what we want to build the structure for. We don't have the structure for the green one. What kind of things could happen by just copying the coordinates of the purple that would go wrong? Or that would tell me that the model is not good? You think about anything?
Maybe there are hydrophobic residues outside.
Yes, so we would have things that somehow don't map the energy criteria. So we could define a simple energy. The simple energy could, for instance, be, I know that this side here is obviously exposed to solvent, to water. So this should not be hydrophobic, at least as an interface. I will accept some hydrophobic residues, but if the whole thing looks hydrophobic, then maybe I have a problem. Yes, that's one thing. Anything else you can think about?
Problems or
that is another issue. So say that these two here are relatively small residues, and you replace them in the human protein by very large residues, then essentially you have a clash. The model implies that two residues essentially occupy, or the side chains of two residues occupy the same position. And that's impossible, right? So another way in which you could see that is very similar to the hydrophobic. It could be that you have two positively charged residues near each other. On top, maybe in the fly protein, that was a salt bridge, so it was a negative and a positive charged residue. So there are all kinds of things, of violations, simple violations, in which you can compare the model. Can say that the model is not good. Now the question is, how can you, if you see that the model is not good, what can you do?
You can run a relaxation simulation.
Minimize the energy.
Yeah.
Exactly. That's your second part. So now you can try to correct. And trying to correct, there are different ways in which you could do that. One is, the simplest one is you try to, when you have an immediate clash, you move it a little bit away from the clash. But then very soon you get into a minimization problem. Because maybe you have the positive hit, the hydrophobicity hit, maybe one moving a few out of would already help. Maybe that would lead to new clashes, and then you're essentially exactly at the MD kind of simulation. The refinement process here. Now, there are different problems on different levels. Let me just quickly go through a few of the standard models. The one is Modeler from the André Charlie lab, who did his PhD at Birkbeck with Tom Blundell, then the postdoc at Harvard with Martin Karplus. He did MD simulation. So he started comparative modeling here with Tom Blundell, got into the refinement kind of part of comparative modeling, the MD simulations at Harvard, went to Rockefeller University and built up his package, and later to UCSF. Even 5 years ago, over 400 publications. Today, his most important work is really trying to put all of many tools together to fit into low resolution maps, or fit different experimental constraints, including modeling. So this part of Modeler here is one aspect, but Modeler is one of the most used methods for comparative modeling. These are the two original publications. The person who came later is Marc-Martin Renom. Modeler is one of the two major packages that puts out over 30 million models through a website. That's the interface. And its pipeline is essentially what we said before. You find the fold, you align the template in the target, you build a model, you assess the model, you ask whether the model is okay or not. If the model is not okay, you either try to see whether maybe with a different template, the same process would have been different. So a different template, simply one way of doing that is you find the template that is the closest in terms of sequence alignment. But maybe that gets more clashes than the second closest. Okay? So this is the reason for this pipeline here, or for this step. Another way is you could try, yes?
Does it compare the first model with the refined model?
Yes. So the pipeline is optimized such that this reduces, unless the condition of satisfaction is reached. And if it does not improve, then it will find whatever was best at the time. Now, the other idea here is that one of the problems, and implicitly what Jade said, this is part of the story here, to learn how to do the alignment right. So you could, in fact, try from the incorrectness of your model, try to see whether maybe there's something you can change in the alignment. So not sort of optimize the alignment method, but turn the alignment method in some sense upside down. So do something that is not optimal on the alignment method, but that would slightly change the alignment. Maybe there's something that in fact does help to do a better model. And you immediately see that this is sort of can be very costly. It's CPU costly. So one simple criterion for energy is constraint satisfaction, and the simplest one is this one here, the C alpha distance. So that essentially is the observed data, and you try to simply put your model into this observation. And that's this sort of the simplest energy. Long before you are having missing loops, you want to see that in fact the structure, proteins are not very bendable. They're not like rubber bands. They are very stiff. So you can hardly not do that. I mean, you see that the distribution is fairly sharp. Okay, so if you're very far away from that over the entire model, you're very wrong, and you try to change that. You try to change that, and this is just one run of such an energy minimization. You minimize with a variety of things. Again, I'm just rushing through. I do not expect you to know any of these in the exam. In fact, I hope that I expect you to know less than I say. But you get my point. I just want to still flash it in front of you to have shown you that underneath, in particular, I'm going to say a few things in a moment, underneath there is an immense amount of complicated computational biology done and intelligence put into this. Ultimately is rigid bodies. The reason why it's rigid bodies is because otherwise the problem of optimization completely explodes. So this is still not could still not be relieved. Modeler does multiple models. Multiple models I said, look at a different template if the one didn't work at all. But another aspect of doing multiple models is you sort of cut and paste them together. So you say maybe here this one is best, here this one is best. So you take different elements and see whether that helps you in the constraint minimization. Typical problems here are a side chain packing. You get the backbone slightly wrong, you misalign the sequence, and you get a totally wrong template. Now if you get a totally wrong template, why is your energy not showing you that? So what I'm saying, typical mistakes, means those, in fact, after the modeling pipeline, come out as the best prediction of Modeler, or any other package. Modeler is clearly one, if not the best, one of the best out there. So this is the kind of thing that the energy minimization did not recognize, where the energy minimization did not recognize the wrong template.
Because there would be like, I don't know, like energy landscape, there would be like several minima, right? They might get to the wrong one. And
oh, that's an interesting one. And that gets us to this image here. RMSD error, low is on that side, high on that side. The dope score, or the somehow you can think about it as energy, where low energy is here. And you see that for some, the lowest energy state, in fact, is the lowest. This is the closest structure, while here the lowest energy state is not the closest, so there are a few that are closer, and this is a little bit more strongly the same as here, although in all of them, essentially low energy solutions are also very close in terms of the model. Your image of different minima is somehow, here's an example for that. This is somehow applicable, but could you really imagine that a protein that is totally different, so different fold, creates an energy situation almost equal? Okay, let me turn that around. I'm going to ask you, if I took a simple molecular dynamics function, and I took an all beta protein, and I put it into an all alpha structure, and I ask the energy function, are you happy? Would you not assume that in most of the cases the energy function will say, no, I'm absolutely not happy. All alpha does not fit all beta. Is that what you expect? If that's what you expect, you're in it for a surprise. This is not the case. Most energy functions, the simple energy functions, cannot solve that problem. In fact, all beta fits perfectly according to energy models into all alpha. What molecular dynamics can do is to say this or that, so we are very close to the real solution. If we move it a little bit away, we get back to the real solution. They're extremely good at that. But if you are in a universe where you're completely wrong, it can look quite all right. So it looks like a similar minimum, although it's something totally different. And there is no good solution for that problem. So it does, in fact, happen. Now, what if the loop is missing? So if you have essentially another part, so your query is a human protein, the human protein is a little bit longer, you have 5 additional residues. What can you do then? How could you model those? So, let's just, here's another example. So here we have an additional insertion. So this is essentially the corresponding points are here. This one goes here, but here you have a longer loop. How can I model that? So that I cannot model it from that structure because the parts where they are clearly mapping are here, but then this one has a shorter one and this has a longer one. How can I do that? There are 2 ways in which you can do that. Tell me the simplest.
I'm just going to have to go with hands.
Oh, you're just going to say whatever random confirmation, all are possible? But there are many ways. And in this particular case, 1, 2, 3, 4, 4 residues. There are many ways in which you can put them. Why put them this way? Why not this way, not that way, not?
Maybe test the lowest energy and no clashes.
So you compile an energy. So you do an energy minimization for that one. That is one way of doing it. And this is what I refer to in the more complicated version of doing it. What's the simpler version?
You can do an empirical analysis of all known loops.
Yeah, you go through PDB. You know the sort of begin and end point. You find in PDB everything that has a similar begin and end point has 4 instead of 2 residues. And then you just copy that. Now you can still do the energy function, but now it's a much simpler one. You only distinguish between one that looks like that or looks like that, and it's in its local environment. That is the historical way, and if that doesn't work because there is nothing in the PDB that looks like it, then you do energy minimization, and that is this one here. So that's exactly what you guys already said, molecular dynamics. This is a slightly outdated figure from the Charlie lab from Anders Fischer, where he shows the loop length versus the RMSD, so increasing difference between the loop that you do by molecular dynamics, that you do suggest by molecular dynamics, and the actual one in the database, the experimental structure that is, number of residues from 0 to 14. You see that the RMSD stays below 2 for roughly until 8 residues. So meaning, if you said that for a loop length of 8, 2 angstrom is still a deviation that you accept, then essentially you can model it. And at some point it explodes here, as you see. It's difficult to say where you clearly cannot do it anymore, but for 10 it becomes very tough.
What is the resolution of current crystallography, for example, for 10 residues of a loop? Because I think loops are always quite bad resolution.
Good point. Good point. But good point. Very good point. Ultimately what that translates to is that the field at about the time of 2000, in most of the cases of 12 residual loops, could not distinguish between that and that. And something, so between confirmation and something completely different. And that is not the resolution of loops in the database. So I'm not entirely sure where this number started. Maybe it was 14, but at some length of the loop, those days, clearly the field was nowhere near it. Today, the difference that has happened over the last 15 years is we can push this a little bit higher. So 14 residue loops we sort of can do most of the time, essentially relatively right within the resolution of the experimental structure. But those days that was not the case. Another model, model building software, a Swiss model, today done by the Schwede lab in Basel, this Thorsten Schwede, who actually started as an X-ray crystallographer. Originally it came from Manuel Peitsch, Nicaragua, and the original model published here had that's the interface. Again, they published over 30 million models. The initial idea was, from Manuel Peitsch, have something that is totally automated. Modeler and all the other modeling packages at the time were not. They were for expert users. For non-expert users, something that can actually dump structures on a website directly for others to look at. And the philosophy was do less, then you do better. The less you do, the fewer mistakes you make. And that essentially was the end. It was really a philosophy decision. Manuel Peitsch had been working on comparative modeling for many years and had a lot of experience. And he condensed all that experience to say, the less I do, the better I'm off. And essentially the way the model proceeded is alignment by blast or psi-blast to find the template, the protein in the database of PDB that has a similar structure, copy the coordinates, stop. No refinement, because in fact that was the first one who dared to do that like that. And as you can imagine, the whole field killed him. And then it was for the first time that the community at large tested it. It turned out that anybody who refined did worse. So all the intelligence that went into refinement led in the end to worse solutions. has slightly changed. And it has also true that today Swiss model no longer does that. Today they have a pipeline that is very similar to the modeler pipeline. Some of the slides from Christopher Wilton. But I'm going to rush through it. The begin here, as you may observe, is no longer PSI-BLAST, but HH search. So we're looking at pairwise sequence alignments, at sequence profile alignment, and as profile alignments, and they are all looked at simultaneously, bringing up different models that are looked at simultaneously. So not only to go in the pathway, they use substrates. That is an important part, whether the two proteins are known to bind similar substrates, and this could actually lead to using a template of a different structure simply because it has a similar binding mode that is known. They look at prediction of secondary structures, transmembrane helices, and other aspects, put all of that into building the model core, do loop modeling as I showed you before, do the side chain again by some energy minimization, and then energy minimize the whole thing. At the end of the process, you have a model that somehow looks like that. In some cases, you have not so well-defined structures. And you have, in fact, an energy function here. That is the residue number, so protein residue 50 to 350. If all of these lines that you see or don't see were 0, then the energy would be absolutely happy with the model. You see that by different scores, per residue, average over helices, average over secondary structure segments, all of these deviate from 0. That means the final best model is not the model that completely satisfies all your constraints. You simply could not. That means that, however, this function here shows you something that, yeah, there are regions of the model where the model is much more reliable than others. And in some sense, if you want, this points to movement in the structure or to inaccuracy in the structure too. So we can overlay structure resolution on top of that. And we'll often see that where the template that you used, the structure, the experimental structure, has more mobility, so less well-resolved structure, that is also where this energy function goes up. So maybe the solution is not necessarily wrong, it's just that we have movement in that area, right? So the constraints are not completely satisfied because the protein moves. It is automated, increasingly comprehensive and flexible. And today, so in the talk where I talk about comparative modeling in much more detail, I will tell you that there are other methods out there. HHPRED does something like that, but it does a much better part on doing the alignment. Okay? 3D Jigsaw simply assumes that every sort of local structural motif is already somewhere in the PDB, and all we need to do is find these motifs and then sort of string them up and essentially build structures from the known motifs we have. That's the idea here. What if is really a method that is optimized on visualization of the comparative modeling building process. And the reason for the name here is that what if I see the clash at this point? What if I had two positive charges? What if I could move that? Would that change the binding affinity? Would that change the model? Would that improve the model? So it's very oriented toward users who want to move things on the screen around. Coma is another method in some sense like this one here that tries to improve the alignment method. And there we are. "30 slides later. How would you assess prediction performance? So you get some method that is comparative modeling, and you get a certain performance, or you get a certain model, and then you assess your performance by looking at the 3D structures you know. Is there any way in which you can do it better? Or in which you can, so I made a mistake at the way I strung these slides together. Okay, let me just go back to another slide that will come later, because I made a mistake in stringing them together. Let's get back to the issue underneath. The protein structure prediction problem. That is the following. I give you a sequence. I told you, I take a sequence, I throw it into a solvent, it adopts a unique three-dimensional structure. There's nothing in there that has to do with biology. It's biophysics. If the protein can do that, we should be able to do that. And indeed, in the 60s, there was a headline publication in the Washington Post that said the protein structure prediction problem has been solved. Okay? There was, so in the 60s there was there was the headline, and that was the Washington Post. In the 70s there was one in the New York Times. And in the 90s there was one in the Washington Post again. So what you already see here is this could not have been right, because then this one would not be right? So we cannot solve the same problem again. So both of them are obviously wrong. And I already told you that the 91 is also wrong. Why? What could have happened? And one thing is absolutely clear. The people who are underneath, so this was peer-reviewed work, so the newspaper headline came after the peer-reviewed work. And in all of these cases, the people who had been the authors of the methods that allegedly solved the problem were very intelligent, clever people, absolute stars in the field, who have changed the field, have impacted the field importantly. So they were not idiots. What do you believe went wrong? Let's begin with the first one. Any idea what could have gone wrong in the 60s? Do you know anything about 60s and protein structures?
I mean, it's like the time when they first figured out how to crystallize protein.
Exactly. There was the time when they had the first two structures. Yes. And somebody had predicted the structure. They looked like one of these two. There's two, I will show you some images, happen to be both alpha helical. And they looked very similar. So somebody optimized a method to get these structures, and it didn't work when new structures came forward. As simple as that. It sounded very reasonable for whatever they had. In the 70s, this is still easy. In the 90s, actually now we have 100 structures. So that is no longer quite the explanation.
Maybe in the 90s, like biologically doesn't make sense, all those predictions.
Sorry? No. So I'm asking, so again, we can make the same sort of argument why it went wrong in the 60s and in the 70s. Because there was not enough experimental data to see that what the people had developed were methods that were specific to the structures known. They essentially overfit the parameters. In the 90s, we had 100 structures. So overfitting the parameters already was more complicated. Why could it still go wrong? It's actually remarkable how much you can overfit. 100 is a small number, is the answer. On top, the method in the 90s couldn't actually predict for all the 100 structures. It could predict only for a very small subset, namely the shorter ones that were rich in certain bonds. And of those, there were actually not that many left. And back to essentially the story of the 70s, they overfit, and they did not realize. None of those is trying to cheat here. Science is full of people who cheat openly and succeed with that. Those are not the cases here. So that brings me back to that question. How can we avoid that?
You can use unpublished experimental structures which you predict in the first place, and when you get the experimental structure, you compare the two.
That's essentially the idea of CASP, the critical assessment of structure prediction. So what it does is the organizers of CASP collect experimental structures, or essentially not structures really, collect things for which structures will be determined soon. So essentially they talk to experimental crystallographers mostly, ask them, what is it that you're going to work on? What is it that you believe we will have published by some point in late summer? Those are put out for predictors. They're 3 months to predict. And then they are assessed, and then there is a meeting to discuss how well the results work. It started with John Mould and Tim Hubbard. So the beginning there's a team behind it. Christoph Fidelis is still the only one that is left. Today, Christoph and Andre and Anna, major player, and Thorsten Schwede, who was already there. So the first result is there's no successful prediction of 3D structure from sequence yet. I put this slide up 10 years ago, 15 years ago. It remains true. So we are much further than before, but this still is true. And in fact, the second most important point is really, really true. So we can really not do it. So we have some examples where 3D prediction works. Call is one example. I'm going to show you some other examples. But what is absolutely much better here is the recognition of when we are wrong. So this type of issue that I talked about before, I put an all alpha protein into an all beta protein. This problem we really solve much better today than we did before. And we, in many ways, extracted principles from protein structures. But the most important point here really is we improved homology modeling, mostly because there are more structures in the database. But much, much more is to be said, and much more is better here. Most of the improvement of this one comes really from doing better sequence alignment. That's the profile alignment story, partially. Another big change from CASP was that people saw what works in some other lab, and they used that for their own server. So essentially, they created servers, meta servers, meta servers, and so forth. And CASP triggered a lot of this. And that, in some sense, is one of the major long-lasting outcomes, a positive outcome. Problems with CASP are, well, one is that the way this experiment is designed, you have a limited number of samples. If the number of samples here gets too high, then it gets too much of a load for the predictors. And if it gets too low, you immediately see there's a statistical issue. So say method A uses 53 of the 100 proteins and reaches performant X. Method B uses 35 and reaches X plus Y. Is B better? Well, clearly, X plus Y with Y being a positive number is higher than X. There are a couple of things you have to worry about before answering that question. Can you just throw some at me of those? For those of you who were in the lecture before, this is a theme that repeats itself again and again.
It really depends, right? I mean, it really depends in a way. B could maybe have like a bias. B happens to select from easier proteins? Or
oh, this is the first important issue. This is a very, very important issue. So what if these 35 would have been easy to predict? So you have to really look at the same data set, right? That's one issue. And we get back to that in a minute. Another issue that's very important is the value of Y. So if I said that, say we talked about secondary structure prediction, and say secondary structure prediction, the performance is 70%, X is 70, and Y is 71. Is 71 more than 70? Well, yes, numerically it is. What's the next thing you have to ask? So,
how many true positives there are and how many true negatives? Or false positives?
That is a very important one. And so it could be that one method is only predicting, say helices, in my case of the secondary structure prediction. And you want a method that predicts helices and strands. So this would be another question. This is true. But let's just, I will get back to that issue here next week. No, in 2 weeks from now. There's a so-called Q3, a three-state prediction accuracy in secondary structure prediction, which essentially is helix, strand, other. So how many residues are correctly predicted in either of those three states? Okay, and if the performance were 70%, and that is method A, and B had number 71. and say they would be balanced, equally balanced in terms of helix strand other. So in that sense, false positive or whatever you look at is somehow similar, except for is 1 percentage points more here.
The margin of error, like 1%, is very small.
That is the important. How do you know?
I mean, 1% from 70 is like very small, right? I mean, 1% improvement. I mean, you can't really say anything 1% improvement, right?
Can you not?
Especially using less data.
Which data?
I mean, for example, the 35, if you use 35, you get 71%.
Okay. I'm going to satisfy you. You're too emotional. No, it's wrong. Let me do it differently. I said the wrong thing. B is 77. Now what? Is 7 percentage points an important improvement? You say yes, defend it.
It's like an improvement of 10% of the original.
This is one way of thinking about it. Put it into the original number. And 10% of the original number. This sounds like a lot, but this is not enough. There are a couple of questions you have, or a couple of issues you have to think about before you can actually answer. You were both rushed. Can somebody tell me what a couple of things that we should look at?
Your point was right with the error rate. When you have like an error rate.
What is error rate?
Like when it can be 50% or 90%.
How do I get the error rate? How do I get the error rate? Anybody else? Somebody else? How would I compile an error rate? Where do I get an error rate from? This number 70 or 77. What is that? It's a way the method performs based on some cross-validation data set that you have, right? Say your data set had 100 proteins. Then essentially what you have is you have some distribution, and for method A, the average is 70. The standard deviation, essentially the sigma here divided by the square root of the number, in this particular case would be 10, gives you the plus minus standard error, roughly, right? Which would be 10. So in this particular case here, if the sigma would be 10, it's not an unrealistic sigma, then 1 plus minus 1 would be significant. Which in fact would be close to the 71 that I had before. And if you immediately if you had done it on 1,000 proteins, which in fact you can easily do today, then this sigma for this thing is so small that this 1 percentage point would be statistically significant a difference. Okay? That's one thing that is important. Yes. There's something else you have to ask. And what you said before is relevant for it, but there's something else. What you're looking at is the number 70. So you say 7 percentage points is 10% of 70. But 70, I challenge you, is not quite the right number to look at.
The distribution is also important, right?
That's true. And okay, let me just put down what I mean. So what's very, very important is that you know what is random. What would happen if you used no information, just threw a coin? This, in fact, in this particular case is not 50, because we have a 3-state distribution, so roughly it's 33. They're distributed differently. So the random prediction actually is 35. Okay? So now you're beginning to see. So this is, say, lowest. Now, there's something that is top. What is the top number? Is the top number, my question to you, is the top number 100? And I would challenge you that it is not. Why not? And there's something that Michael said before that's relevant for that, for answering that question. Well, the top number here is the one that's given by the experimental resolution. And in fact, that number is nowhere near 100%. There are different ways in which you can put that, but in this particular case here, this is ballpark 90. Okay, so now what we're actually looking at is the interval between these two numbers. So on the interval between these two numbers, I'm a little bit 55. So on the 55, 7 percentage points is in fact more than the 7 percentage points. And again, so another way in which you could use that sort of, this sort of is double, but this is double plus something substantially more. Plus you have the statistical significance. All of these things, so here I show secondary structure prediction. All of these things you have to immediately address when you ask whether or not Y is an important improvement. In principle, you should ask the next question, which is, what do I want my prediction method for? And is it an improvement for that goal? So I want to have a better model to predict function. Is Y improving so much that I get better function? That ultimately is the question you have to address, but typically in most work you cannot do that. Those things you typically can address. And the most important one that you picked up immediately, you have to really compare apples and apples. So you have to compare the same data sets. In this particular case, I'm just going to tell you what this shows. Don't look at the graph. What I did here is, and this is really based on secondary structure prediction methods, and I wanted to compare a bunch of methods. That is one of the past CASP. This is a realistic example for one of the past CASP, and I wanted to see which secondary structure prediction method is best. I believe they all, so the method that had most predicted on most had 29. Or I believe everybody had at least predicted 29. And 11 of those were identical. So back to the issue, maybe some were easier, some were more difficult. If I really wanted an identical subset for all methods, I had 11. I had 8 different methods. And now the question was, should I compare method on 29 proteins, where they differ, so different 29, or on 11 that they are identical? And again, 11, both numbers are very small, you understand that. But 11 is 1/3. I mean, if 100 would be sort of okay, 29 is much more sort of okay than 11, right? And what the experiment showed is this is much better a solution than that one. With that one, by choosing the simplest 21, every method could become the winner in a CASP. Just essentially I simulated many CASP experiments. And by just resampling from the list that we had, every method could become the winner, although we knew that some methods in the 8 really performed significantly worse. So this, because it seems to be higher, is a bad idea. But you always have to use identical subsets. One shortcut, if methods predict different things, is that you could do comparisons. So where you compare method A with B with C, always on identical subsets. So in this particular case, by these pairwise comparisons, you're not really having gone down, you do not have to have 11 that are identical to all of them. You could have, the subset here could be 25, this could be 22, so those would be different subsets, but then you would essentially rank according to how often do you win against all methods, and you would sort of get a rank advantage. There are all kinds of difficulties with that. This is still, no it's not. Yes, I do want to make that point. Sorry for the animation. I thought I got it out. What I show here is sequence identity. What I show here is RMSD, different comparative modeling methods, 100 to 40. RMSD, 5 is the threshold here. So most methods somehow, as long as you have enough sequence identity, do comparative modeling within the range of what is experimental accuracy in some sense, okay? Models are good as long as you have significant sequence identity. That being said, what this one shows here is the fraction of the protein that you model. So you come in with your query and you have a template, but the query doesn't fit the entire template. It fits only some fraction. The more sequence identity goes down, the smaller the fraction it fits. And you see the realm at which most methods somehow perform okay is in fact one where you model at most round about 60%. Ballpark, a little bit over half here, 60 to 80%. So comparative modeling is by far the most successful method, but a lot is left out. When we say that we can model 30 million models and 60 million sequences, that means that it is a fraction of all these. In every single one, we essentially model a fraction of a domain. This is the best we can do. It's remarkably successful, but it's still only a fraction of the reality that we want to capture. That is what this one is supposed to do. This is back to my theme of models and reality. This is not a pipe. In fact, it's an image of a pipe. The image helps us to possibly even smell the tobacco, but it does not, it helps us to understand certain things, but it's not the same thing. And that's something that we have to remember. And another thing that we have to remember, that you, for anything you do, it's not this one is sort of comparative modeling related, but it's true for anything you build that does prediction or estimate something, a model is wrong. Identify where it's wrong. You can learn a lot about the prediction method, and people can benefit a lot about the prediction method if you can tell them, okay, here's the region where I believe it more, and here's the region where I believe it less. Try to find ways to do that, and try to learn from where it's wrong and where it's right. Thomas Schrader from Swiss Model puts it in a slightly different context, saying that essentially the model is, there is no value in a model. The value of the model really is to understand some data. Typically the data is function. So how much of this model, how much does the model help me to understand the binding site, and how much does the binding site then help me to understand why a protein functions? And if that helps me, how much does it help me to change my experiment and see whether then binding stops? And if it does, then the model was useful. And that is more important than RMSD. That's ultimately the point here. And with that, we are perfectly positioned for the next 160 slides. And I have to find a shortcut because I really want to go. And I want to begin here with the structure by experiment. So the most important source of protein structures is X-ray crystallography. For an older PDB, about 89% of all the PDB structures were done by X-ray crystallography. In order to do X-ray crystallography, what you need to do first is you need to express your protein in a high concentration, and you need to so purify it to very high concentrations. Bless you. Why do you need such a high concentration? This is simple. The answer is on the slide, and it's very simple.
So you wouldn't have the diffraction pattern?
That is correct, but there's a step missing. But now I give it away, huh?
You need the crystal.
Crystallization. In order to grow a crystal, the way you grow crystals, and this is not only for protein crystal, I mean any crystal, is by you increase the density. You put something at a very, very, very high density. Now, when the density is high enough, you sort of need to lead to the point of a phase transition, and then a crystal grows. And that is exactly what you have to do here. So high mass, highly purified density of protein. Once you have that, you shoot synchrotron beams. So normal X-ray beams are not enough. So you really need synchrotrons. So the big beams. And from the diffraction pattern, what you get is an electron density map. Density map means you have a probability of where the electrons are. All right? And that typically on graphs is shown as a blue haze, and I'll show you some of these images on the next slide. And from that electron density map, you create the atomic model. How do you create the atomic model from the electron density map? You just thread it in. You just ask, how well does your residue or your atoms fit into that map? You know the sequence, you just put them in and see how they fit. Okay? How the side chains orient themselves. And the orientation of the side chain, okay, now we are at the point where I would have to make it much darker. 3 angstrom, 1 angstrom. So in the 1 angstrom, what I assume you hopefully can see is that the blue really fits the green here. The blue mesh. So that's the electron density map. And maybe you can even see that in here is black. So in this particular case, you can even see that the ring has a hole in the electron density map. Well, this one you cannot see, but maybe what you can see is that the mesh, the blue mesh, sort of extends over the entire place here. So you clearly cannot distinguish between different orientation. You clearly can put this in different ways, and you can move in this direction. That is beginning there, you cannot really see it anymore. But you can actually move it in that direction, you can move it in that direction, and you can clearly move it in that direction. So 3 angstrom resolution is one that is already not accurate enough for all the side chain placement. Most of the PDB structures are today, very good structures are here. Still some are coming here. Very few are on that level here. There are some in the PDB now. There are still some on that level. So this is still a high resolution structure. NMR. In NMR, the big advantage is you do not need the high concentration. You do not need to grow a crystal. You can essentially put the protein in its native solution or solvent into a pipette. And then you need a magnet. You excite the protons, and you essentially measure how the protons fall back. From the protons falling back, you see correlations, but you essentially see distances. It's a little bit complicated to find the final model. So one complication of this is in the olden days was one of the biggest NMR machines. Today it's relatively normal. This is the New York City College. It's a large room full of these machines. The dimension of this machine, so that's a person up here. And that machine is so big only because it's a big magnet. There's nothing else in there. It's just a big magnet. And this room is such that if you walk in with any credit card, this is a gutter. In fact, this is a Faraday cage, so you completely isolate that from the environment. That is a major piece of equipment, a building. This is expensive to build, these machines are expensive, and they're complicated to operate. And then the problem is really that you have to also assign the model. What you see here are different NMR models. In some places, all these models, 1, 2, 3, 4, 5, 6, 7, 8 or something models, 20 models, they fall in the same place. On some they don't, and on some they really don't. These different models are simply, in a computer, you fit the model to the experimental data. So essentially, we are having the same situation that we had before. We have a 2D map, but the 2D map is not complete. You do know the constraints, but you do not know whether it's the cysteine at position 16 that binds the cysteine at position 60, or is it the cysteine at position 120. So which one? That's an ambiguous constraint. You know, there's a cysteine-cysteine bond, but not exactly which one, because you don't get that out of the NMR spectrometer, the assignment. So you essentially try different ways of fitting the structure into the constraint that you have, and what you see are these 20 different fits. Do you know how NMR spectroscopists interpret this image? So the positive interpretation here is that what NMR measures, because we're not talking about a crystal, measures is function. We are seeing the motion in solvent. What we are seeing is that there are regions in the protein where you have no movement. And then there are regions, particularly here, where you have a lot of movement. That is functionally relevant, as is the rigidity of this one. Okay? So the positive interpretation of the different NMR models is they show you something about motion. They show you something about function. The negative interpretation is the constraints are not good enough to give you the real solution. Essentially, the different possibilities indicate one of them is right, you just don't know which one. Okay? And I believe there are many, many NMR structures that are examples for it is wrong. And there are many where you really, in careful detail, see there's motion visible in there. Both is true. But going back to the, I'm not entirely sure why I didn't have that before, but the NMR thing is big. The synchrotron here, this actually, they are accelerated here. This is Illinois. Here it's in CERN, and you see now we are talking 27 kilometers. This is just one of those views of the tunnel. These are huge constructs of something that you need in order to get a single protein structure. Okay, why not look at it with a microscope? Well, actually you can do that too. Not quite as easily as an EM, as an electron microscope, and it's cryo, so you have to freeze the protein. Still not a crystal, but it's essentially taken out of, not taken out with its, frozen with its solvent, and you hope that upon freezing you don't change a lot. EM currently, and this is clearly not currently, this is outdated and this is absolutely changing, made up 1% of all the structures. This number is increasing rapidly. Historically, EM got to resolutions in that ballpark here, some very good ones in that ballpark, very, very, very few in that ballpark. Now, with modern EM, so now you can build microscopes, you can buy microscope for ballpark of 5 million that give you something that is this and better. And that possibly is the sort of way in which structural biology will be done in the future. It's still not that cheap, still not that simple. So in order to get that, the problem that you now have is essentially these things make about 100,000 images of the same protein structure. So essentially you put it into a film upon the carrier, upon a glass plate, and then you rapidly shoot a lot of pictures, and you essentially get 100,000. And then when you put them to compare them, you realize they are different. So now do your thing. That's the challenge at the moment, right? Which one is right? Because essentially the protein over the course of the duration of these 100,000 images simply changes. And you have to sort out what in this is just motion, what is noise, what's the common theme, what is the real structure. But with that carefully done, we get in the ballpark of below 4 angstrom now. Now, even the high resolution map could help you if you had to compare it. For in this particular case, this is a slide shown here, but the group of Andrei Shali is doing a lot of work in this direction too, where they use modeler to get the units, and then they put these units together and fit them into the EM image. And Baumeister at the Max Planck Institute here in Munich is in fact working on the same thing, because he's very good at pushing the EM story, the microscopy story. So again, this is something that is going into the future. So structural genomics, let me just talk about the idea, the universe of all the proteins. Some of these, so the structures are islands somehow, and I already told you that the islands are a little bit so smashed out, so the water levels between islands is very shallow. But now the idea of structural genomics was find all the islands where, and for this I have to get back into the original, find wide islands. So find proteins for which we do not know the structure, or any structure, and do a structure for those. For all the others, apply comparative modeling, do not do another structure, although it may be important, but let's just fill out all the universe. And this effort has been handled by many consortia with a lot of money. This was funded for 15 years at a level of over 150 million per year, so there's some major funding. They built up pipelines, target selection, so first the question is which one should we go for? Then cloning, expression purification, crystallization, X-ray diffraction, and structure analysis. And in some sense, the few of the assumptions were that ultimately we can do a structure for $100,000. When structural genomics started in 2000, the price tag was more closer to $1 million. So the hope was to sort of cut it down by a factor of 5 through this investment. To automate structure determination, the assumption was crystallization was a major bottleneck. So this actually happened. It was automated. And this initiative did a lot to do this better. The assumption that crystallization is the major bottleneck turned out to be wrong. Essentially every single step on the path is a major bottleneck. We have to determine about 1 to 10,000 structures in order to sort of fill out all the white islands, was the assumption. That turned out to be entirely wrong. Structural genomics determined about 5,000, so half of that, or depending on how we count, maybe even 10. But we are nowhere near all of it. The assumption was that prokaryotic biology is enough to get to those. The background here is it's much easier to do a structure for a prokaryote from everything. So it begins from the sequencing, gets down to the expression, purification, crystallization. Every step of the way is 10 times easier for prokaryotes than for eukaryotes. So the hope was that we do it for that all the islands have a prokaryote on it, and that then essentially the rest we do by comparative modeling. And that also turned out to be very wrong. So another assumption was that X-ray suffices to get the main skeletons. Is also not quite true. And that started in 2000. We get the whole story in 10 plus years, and that was absolutely not true. But one story here I want to show, it's a slide in between. Shows what we refer to as the attrition. So 6,000 at that point were cloned, 3,700 were expressed, 1,600 purified, and 200 led to structures. So you start from 6,000, you get 200. And the major point that I want to make is that, crystallization is not on this one here, I'm sorry. But the major bottleneck really is not crystallization, it's just a funnel. Every step has a sort of similar reduction, and every step has to be addressed and improved equally. And so let me just get to the, I'm sorry for my overload. Do not deliver on my promise. There will be a few more slides. What you see is the 3D structure, or some simplification of the 3D structure. And what you see here is a cartoon. What determines secondary structure formation? So you see helices and strands. Do you know what determines them? Why they are formed? Stabilized by hydrogen bonds, and that in fact was the major person behind that is Linus Pauling, who is one of the few people you may know has 2 Nobel Prizes in separate areas, the biochemistry and chemistry and peace, stop the nukes. Here the original work in 51, and in which he essentially predicted that helices would form according to his understanding of sequences. And that in fact the other one would be the pleated sheet. That's the sheet and the helices. These pictures, by the way, that's Pauling, Robert Corey, and Branson. Those were the 3 in the original publications here. This one, and that's a paper from David Heisenberg. The first structure, as you already observed, was later. Kandrew and Perutz, essentially they worked on this for both groups, both people, both teams worked on it for more than a decade. This one was 58, this one was 60. Both got Nobel Prizes for that. And you see, again, this is a duplication of the same thing. Myoglobin, hemoglobin. The reason why they picked that is because they could purify and express it relatively easily. They could relatively easily get it large amounts. And as you see, these both have one thing in common, not the word globin. Yes, they're both globin folds, but is there anything in terms of secondary structure that they share?
They have no
Sheets. They have no strands, no sheets. Strands is one. When two strands come together, it's called a sheet. Yes, they don't have that, and in fact, the first 4 structures were all helical, and therefore that the first 4 structures took until the 70s to do. So for a long time, Pauling was supposed to be, or was thought to be wrong with this idea of beta strands, because they only observed helices. Now, how could you assign secondary structure from 3D structure? So go from that to that.
There is like a regular pattern in every helix where, every
Geometry.
Third or fourth base is like in a specific position, so you can calculate the coordinates where it should be to form a helix.
You're mixing 2 concepts here. You actually name 2 different methods that use 2 different principles implicitly. It is true that what you show, I believe the first one is you could sort of see that there is some geometric pattern. If it works like that, that's a helix. If it works like this sheety thing, then it's a strand. Another way of doing it, and that's the I and I plus 4, is simply the idea going back. That's DSSP from Chrysander and Wolfgang Kapsch, and that is going back to Pauling's idea. You begin by defining a hydrogen bond. If secondary structure is formed by hydrogen bonds, then why not look at the hydrogen bonds by a simple energy function, decide where the hydrogen bond is, and then look for patterns I and I plus 4. Then, if you see these patterns, I plus 4, I plus 2, and I plus 5, no, 6. When you see these continued patterns of hydrogen bond formation, then you see it's a helix. If you, on the other hand, have I and I plus 2 binding to J and J plus 2 or J minus 2, then you call it a sheet. And if it's plus 2, it's a parallel sheet. If it's minus 2, it's an antiparallel sheet. Right? So like this or like that. And in fact, in this particular case here, we have both cases. Here, these 2 are parallel, and these 2 here are antiparallel. So oriented like that, and this is what you immediately actually see in these arrows, the orientation. There are many other assignment methods. And let me just say that's it. The one that is most used today still is DSSP. I believe it's the only one that really is used. And I believe I leave it here and leave it open for questions. Thank you for your attention.